So far, we have developed our understanding of the plant phylogeny as nested clades, in which at every node or splitting point, there's usually one minor branch that retains the primitive state, and the other major branch becomes the main lineage that bears some new innovation and gives way to more nodes with more splits. Archaeplastids split into minor rhodophyta and major viridiplantae. Viridiplantae splits into minor chlorophyta and major streptophyta, and so forth. Once we get to the original land plant, most recent common ancestor of the kingdom Plantae, there's actually a grade of 2 or 3 basal lineages, giving rise to the liverworts, the mosses, and the hornworts. And sometimes we refer to these guys as the non-vascular plants, although this categorization doesn't reflect evolutionary closeness with each other. Hornworts, for example, are further out on the tree relative to liverworts and mosses, and therefore a hornwort is more closely related to a banana than it is to a moss. In lab, you'll be treated to a buffet table of whatever we're able to collect locally of live plants from these lineages, and you'll learn their fancy names. Liverworts are the Marcantiophyta, mosses the Bryophyta, and the hornworts are the Anthoceratophyta, although we usually don't have any hornworts in our lab display. In addition, you'll be making microscopic observations on prepared slides of mosses and ferns, distinguishing the different gametangia on the gametophytes of these plants. More details about that follow in the next lecture, in which we'll focus on alternation of generations and all of the associated terminology. The major lineage emerging after these basal minor lineages split away is the Polysporangiophyta, plants that have multiple sporangia on their sporophyte stages. And the major branch of the Polysporangiophytes is the Eutracheophyta, plants with true vascular tissue, that is, reinforced tubes conducting water, minerals, and nutrients. In the book, there are some photos of some really amazing fossils that recognize some of the basal Polysporangiophytes, the ones not in the Eutracheophyta because they lack true vascular tissue. All such plants are extinct now, but Aglaophyton and Rhynia fossils show exquisite microscopic detail, which is how we know that they lacked true vascular tissue, even though the plants themselves died in the early Devonian. These plants come from the Rhynie chert in Scotland, preserved under unique conditions for perfect petrification by silica-rich hot waters arising from volcanic springs. These fossils even show associations between these earliest of plants and mycorrhizal symbionts. Well, anyways, these Rhynie plant fossils are all Polysporangiophytes, but not Eutracheophytes. Today, in contrast, the only Polysporangiophytes still in existence are the Eutracheophytes, the true vascular plants. So, the layout is going to go like this. The whole clade of land plants is the kingdom Plantae, also called the Embryophyta, a name that will make sense in the next lecture. Mosses, liverworts, and hornworts are the early splits forming a grade of non-vascular embryophyte taxa. The Polysporangiophyta had some early non-vascular members like Rhynia, but is represented today only by the truly vascular plants, the Eutracheophyta. Eutracheophytes split into the Lycophytes and the Euphylophytes. Now, Lycophytes include vascular plants with linear leaves, the ancestral state in which each leaf was served by a single unbranching vein. Flattened leaves with multiple or branching veins was the innovation of the other major clade, the Euphylophytes. The Lycophytes were highly successful during the Paleozoic, though today this group is relatively unimportant and represented by only the club mosses, spike mosses, and the quillworts, all three fairly obscure little plants, and there aren't a whole lot of interesting things to say about them. We may or may not have live material to look at in the laboratory. The Euphylophytes, literally plants with true leaves, is the other group, splitting into the minor group, the Monilophytes, which are the ferns mostly, and the other group, of which spermatophytes, plants with seeds, is the major lineage that we follow up on in a later lecture and lab. This is the group with gymnosperms and angiosperms. For the remainder of this video, I'll cover the chronology of sequential developments in the first half of the Paleozoic as plants made their push onto dry land. The first 3 periods of the Paleozoic are Cambrian, Ordovician, and Silurian. And through these times, we know that plants were gaining their first foothold onto dryland habitats, But our picture is far from complete. These early plants didn't leave us much in the way of fossils. The earliest plant fossils, as I mentioned before, are just fragmentary bits with just enough information to tell us that the plants were definitely in existence no later than the Ordovician. Well, the geologic record from the Cambrian, and actually most of the Ordovician, gives us really no indication that plants had made their way onto dry land. The Silurian gives us some better fossils of early plants, and even one nice example, Baragwanathia, of a purported lycophyte, which, if correct, would mean that true vascular plants, the Eutracheophyta, had already come into existence no later than the Silurian. The Rhynie chert organisms are from the early Devonian, after Baragwanathia, but none of these plants were Eutracheophytes. The story here is that even though Eutracheophytes had originated in the Silurian, the dominant forms of plant life in the early part of the Devonian, at least in Scotland, were still non-vascular. Now, the second half of the Paleozoic consists of three periods: Devonian, Carboniferous, and Permian. Through the Devonian and Carboniferous, the rise of plant life on dry land caused the next great biologically driven change to the atmospheric chemistry and global climate. The rise of forests in which plants grew to huge size and died largely without being decomposed resulted in a massive drawdown of atmospheric CO2 and also an increase in oxygen O2. The carbon dioxide drawdown caused another cold, dry period lasting into the Permian, while the bump in oxygen to perhaps 30%, which is much higher than the current oxygen level of 21%, resulted in rampant, uncontrollable wildfires. There is an abundance of charcoalified plant remains from the Carboniferous, consistent with the wildfire part of the narrative, and also fortuitous to science. Plant material that's been converted into charcoal is largely protected from decomposition, and fossil balls of charcoalified remains can be cut and peeled for microscopic analysis. Not as pretty as the Rhynie chert fossils, but still showing detail at the cellular level. Now, the actual plants that came to predominate in the Carboniferous forests were giant ferns and lycophytes, plants that were vascular but not seed plants. Today, all of the trees making up the world's forests are seed plants, and so these Carboniferous forests would have seemed like an alien landscape to our eyes. The conversion of so much forest biomass into coal may have been due to some kind of shortcoming by microbial decomposers, but it's also likely that the orientation of continents at the time resulted in an equatorial wet belt of intense and continual rains, making the tropical forests of the time similar to a modern peat bog. The rains kept things cool and very wet, and the dead trees would just sink underwater and into an anoxic layer, where the rate of decomposition would be far slower than the rate of biomass production by the growing plants. Now, this is actually relevant to the question of whether or not planting a lot of trees would work as a means of mitigating today's human-caused climatic effects by drawing down the atmospheric carbon dioxide. After all, we know that this is basically what happened during the Paleozoic. As you're thinking about this, consider that the Devonian Carboniferous drawdown occurred over tens of millions of years, in which not only were giant plants growing, they were also not being decomposed. If the carbon fixed in a forest as a result of tree growth is balanced by the microbial decomposition of forest biomass into CO2, then there's zero effect on atmospheric carbon. Back to our plants. The plants that came into world domination during the Carboniferous were largely giant ferns, giant horsetails, and giant club mosses, plants that were vascular but not seed plants. This bit tells us that being vascular was enough for the Paleozoic plants to have enormous success as land plants. They did this all without the benefits of seeds or pollen, both innovations that had not occurred in the ferns, horsetails, or the club mosses. If seeds or pollen weren't part of the equation for these plants' success, then what was? The oversimplified answer is that it was the development of vascular tissue, tubes that could transport water, mineral nutrients, and the sugars produced by photosynthesis over greater distances, allowing the plants to live in drier places by giving them a way to access water below the soil surface, as well as the ability to grow to larger size. The reinforced water-conducting vessels of xylem and phloem, but mostly xylem, provided a physical structure that supported larger masses of photosynthetic cells in the canopy, which then, in turn, required a greater amount of vascular tissue to provide water and minerals, which then would support an even greater mass of canopy. The evolution of vascular tissue basically started a trend of escalating plant size, as well as increasing the plant's ability to persist under drier conditions. There's another positive feedback system going on at the same time, with the activity of plants and the production of soil. Prior to the appearance of plants on land, there would have been no soil, just perhaps the mineral materials of sand and silt and clay that had weathered and eroded from the bare rocky landscape of whole continents. Plants would change things dramatically. The physical penetration by the roots and the acids the roots released sped up substantially the weathering process. And when the plants died, they contributed organics that changed the physical structure and chemistry of the soil, making it better suited to support more plant growth. More plants meant even more soil production, which allowed for even more plant growth. Plant and soil-filled ecosystems quickly replaced barren land habitats. Rainwater, instead of running off as soon as it fell, would be held in place by soil and plant biomass. River flows stabilized, and basically the terrestrial landscape started to look like the world we know, with abundant plant life and even some early animals.